Hi everyone, my name's Melody de Brennan and I'm a PhD student studying the gut microbiome in Brisbane, Australia. This channel is all about science, and what I try to do is take complicated information and distill this into easy to understand chunks that make you understand the world a little bit better and hopefully to see the extraordinary in the ordinary. I'm in my third year of PhD and definitely don't have time for a YouTube channel, and so I've decided to start a series based on what I'm reading as part of my PhD, and it's going to be called The Digest, because I study the gut microbiome. I think that title is hilarious and I would love to take credit, but all credit needs to go to Chloe Yap. So let's get started. The first article we're discussing is called Bioaccumulation of Therapeutic Drugs by Human Gut Bacteria, and it's by Klünemann and alumni. So this article investigates how bacteria might be modifying commonly consumed therapeutic drugs, such as those used to treat depression, anxiety, and diabetes. This is a really great article, and it's probably one of my favorite articles that I read last year, so I'm really excited to share it with you. So what do we know already? Well, we know that therapeutic drugs can significantly affect the gut microbiome, but also that the gut microbiome can modify the effectiveness of therapeutic drugs. Once we consume an oral drug, this drug is then subjected to all of the digestive processes that our food would normally be exposed to, including stomach acid, digestive juices, pancreatic enzymes, bile acids, as well as being affected by the gut microbiome. Gut bacteria can affect drugs in many different ways. They can reduce drug availability by transforming it into something else. They can inactivate the drug, or some drugs actually require gut bacteria to modify it to go into its active form. And some drugs can even become toxic when modified by gut bacteria. Most interactions between drugs and bacteria are thought to be through a process called biotransformation, but this study proposes that there's another factor at play called bioaccumulation. Biotransformation is a process where a chemical is modified by an interaction with a living organism. So a really good example is bacteria fermenting the sugars in milk to make yogurt, or yeast fermenting the sugars in malt to make beer. Bioaccumulation, contrastingly, is the buildup of chemicals in a living organism, and you may have heard of this in relation to fish. Sometimes, due to specific types of pollution, mercury can enter a freshwater system and can work its way up the food chain into fish. When humans then eat that fish, they acquire the mercury faster than the body is able to get rid of this, and this can eventually lead to mercury poisoning. So back to the article. The first thing they wanted to see was whether there were interactions between therapeutic drugs and bacteria. They looked at the interactions between 15 human therapeutic drugs and 25 strains of gut bacteria, which equated to 375 bacteria to drug interactions. They grew each bacteria in growth medium, which is just a liquid that contains the nutrients necessary for the bacteria to grow, and they added a specific amount of each drug to this medium. Once the bacteria had grown, they then collected the liquid that the bacteria was grown in and would check whether the level of drug that they had put in there initially had depleted with the bacteria's growth. They found that 10 out of the 15 drugs they tested were depleted with at least one bacterium, and that all bacteria were able to deplete at least one drug. They took a step further though, because they wanted to know, well, what happened to this drug? So they wanted to see whether it was being biotransformed, that is changed into something else, or whether it was being bioaccumulated, that means accumulating in the bacterium. And they actually found that when they compared the liquid alone to the total culture extract, that's everything in the tube, in 17 cases, The liquid didn't contain the drug, but the total culture extract did. This means that the drug was actually staying unchanged in the bacterial cells, meaning that it was being bioaccumulated. In the other 12 drug bacteria tests, it was thought that this was being biotransformed. However, all bacteria had the ability to both bioaccumulate and biotransform various drugs. They then wanted to find out whether bacterial metabolism was affected by the drugs. Metabolism is defined as the chemical processes that enable life and normal functioning of an organism. Because bacteria were transforming or accumulating the drug, they wanted to know whether this transformation or accumulation was affecting these regular life processes. They found that many of the proteins that were interacting with the drugs were metabolic proteins, and so the authors thought that this indicated that these drugs would be affecting the metabolism of the bacteria. They then wanted to know whether these metabolic changes would affect the community composition of the gut microbiome. It's important to understand the importance of community composition in the gut microbiome. We know that certain species are associated with beneficial effects, and we know that other species are associated with negative effects and diseases. And so when you modify this community composition, this can have significant effects for the host. And so we really do want to know if commonly prescribed drugs are affecting this balance and this community composition. So to test this, they assembled a stable community of 5 different microbes, and these were Bacteroides, Theta iota omicron, or B theta for short, Eubacterium rectale, Lactobacillus gasseri, Ruminococcus taucus, and Streptococcus salivarius. They then grew them up in growth medium with duloxetine, and also a control community was grown up without duloxetine. And duloxetine is an antidepressant drug. The growth of Eubacterium rectale is actually known to be inhibited by duloxetine. And so it was really surprising that when duloxetine was added to this community, Eubacterium growth increased 100-fold. It was therefore hypothesized that another bacterium in the community was bioaccumulating duloxetine and making something that promoted the growth of Eubacterium rectale. A subsequent experiment showed that when they grew Streptococcus salivarius with or without duloxetine, and then harvested that liquid and grew Eubacterium rectale in it, Eubacterium rectale grew better if Streptococcus salivarius had been grown in duloxetine compared to not grown in duloxetine. However, if you do the same thing but you add duloxetine after you remove Streptococcus salivarius, Eubacterium rectale doesn't grow as well as baseline. This means that when Streptococcus salivarius grows in the presence of duloxetine, it makes something that helps Eubacterium rectale to grow. Now, if we zoom out again and look at the big picture, each of us have hundreds of bacterial species in our gut. And if we're taking a medication daily, what effect could this be having on the community composition of our gut microbiomes? And is this a beneficial effect or a detrimental effect? Or does it even matter at all? The final question that the researchers asked in this study is, does this bioaccumulation have an effect on the host? To do this, they used C. elegans, which is a type of roundworm or nematode that lives in the soil. Duloxetine in C. elegans modulates muscle movement, and so they're able to use the movement of C. elegans to determine how it's being affected by the drug. As levels of duloxetine increase, the percentage of moving worms decreases. And so to test this out, they took one bioaccumulating strain of E. coli and one non-bioaccumulating strain, and they grew these in the presence of duloxetine. The liquid that the E. coli were grown in was then taken away and sterilized and then applied to the nematodes. They found that the liquid from the bioaccumulating strain had a less potent effect on the nematodes than the non-bioaccumulating strain liquid. And this makes sense because the bioaccumulating strain would have been removing all the drug from the liquid, whereas the non-bioaccumulating strain would have left it in the liquid. So it makes sense that when the drug was removed from the liquid, this has a less potent effect on the nematodes than when the drug is left in the liquid. Nevertheless, this does show how a bioaccumulating strain of bacteria could modify how a drug is affecting its host. And that's important for us because we are host to a lot of gut bacteria. So to summarize what the study found, gut bacteria can both bioaccumulate and biotransform commonly prescribed drugs. These drugs can affect the metabolism of the bacteria. Bioaccumulation of one drug in one bacterium can affect the growth of another bacterium. And finally, bioaccumulation of drugs in bacteria can modify how this drug is affecting the host. There are still a couple of questions I'd like to look into further after reading this study. And the first is that most of our research on gut bacteria focuses on the colon, which is the last major organ in the digestive system and also houses most of our gut bacteria. However, most orally administered drugs are primarily absorbed in the small intestine, and this is also true for duloxetine. If most oral drugs are primarily absorbed in the small intestine, then we really should be more concerned about the bioaccumulation and biotransformation of drugs in the small intestine and the upper digestive system rather than in the colon. Many of the species that exist in the colon also exist in the small intestine and other parts of the digestive system, but they can differ in abundance. My research mostly focuses on the colon, and I actually don't know if the bacteria used in this study are also in the small intestine, but I'd be interested to find out. I'd also be interested in knowing how much of these drugs are absorbed in the small intestine and how much reaches the colon where most of our bacteria are situated. Altogether, I think this is a great study and it raises further questions and further opportunities for future research. Some examples are how much bioaccumulating and biotransforming is going on in our gut. Are these processes dampening or changing the effects of our prescription drugs? Should we modify the amount of prescription drugs we give to people on an individual basis based on their gut microbiome composition? So I hope you liked that video, and if you did, don't forget to like and subscribe. I'll be doing some more of these videos in the next couple of months, but I'll also be doing my regular science at home videos. Now, if you liked that video or if you have ideas on different studies I can do next or questions about the gut microbiome that you want answered, please write that in the comments. I love reading your comments, and I'll look through them and see if I can take something from that for next time. All right, thanks so much for watching, everyone. I'll see you next time. Bye.